Hello, I am Nasir Ali Afsar from Al Faisal University, Riyadh, Saudi Arabia. I will introduce to you the fundamental concepts in drug treatment of cancers, also called as the cancer chemotherapy. To understand the nature of the problem, we need to understand what is cancer. A cancer, a malignant neoplasm, is a defined tissue, a tissue that is no longer responding to regulatory cues and behaves abnormally like this. To elaborate more, our tissues are tightly regulated in terms of growth and differentiation. If this control becomes erratic, cancer is the outcome. This slide shows the simplified scheme of cancer development and currently available drug options to treat it. Here we are focusing drugs only. A cell with an accumulated DNA damage ultimately leads to a cancer cell. And then the cancer cell has got certain properties which will sustain it throughout its life and also generate more cancer cells. Number one of them is sustained growth signal, uncontrolled replication, sustained angiogenesis, resistance to apoptosis, and an ability to invade and metastasize to distant tissues. To control replication, we have chemotherapy. To stop the sustained growth signal, we could do this by inhibiting the tyrosine kinases coupled with various growth receptors. And now we have anti-angiogenic agents available as well. But still, there are areas which require attention, and there is no remedy if these mechanisms are predominantly erratic. Now, since you know what is cancer and what are different chemotherapeutic approaches, let's review the classification of anti-cancer drugs. Anti-cancer drugs or chemotherapeutic agents could be classified as cytotoxic drugs, hormones, immunoglobulins and tyrosine kinase inhibitors, and certain others which do not fit in any of the classifications. The main group of drugs are these ones: the cytotoxic drugs, the hormones, and immunoglobulins and tyrosine kinase inhibitors. Among cytotoxic drugs, we have another subclassification of three groups: alkylating agents, antimetabolites, and natural products, which are a group of heterogeneous compounds which are grouped together on the basis of their common origin from the natural resources, for example, plants. The alkylating agents are known to cross-link DNA strands. An example is cyclophosphamide. Antimetabolites either disrupt metabolic activity of the affected cells, such as by inhibiting DNA nucleotide synthesis, or folate metabolism. For example, 5-fluorouracil, 6-mercaptopurines, and methotrexate. The natural products have a number of unrelated compounds as a large group, and all of them have different modes of action. For example, topoisomerase inhibition by toposide, microtubule inhibition by texanes, DNA intercalation by anthracyclines, etc. A number of hormonal agonists and antagonists could be used to treat different cancers. For example, anti-estrogens, if it is an estrogen-dependent tumor like breast cancer. The example is here is tamoxifen, or corticosteroids in corticosteroid-responsive cancers, for example, hematological malignancies. Here, in this case, prednisone. Various molecules targeting cellular growth response are grouped together as immunoglobulins and tyrosine kinase inhibitor. Remember, Not all immunoglobulins are tyrosine kinase inhibitors, and not all tyrosine kinase inhibitors are immunoglobulins either. That's why we use the name immunoglobulins and tyrosine kinase inhibitors. For example, imatinib, which is a non-immunoglobulin used in chronic myeloid leukemia. The second one is trastuzumab, which is an immunoglobulin, and is used to inhibit the growth in breast cancer. This slide demonstrates a simple scheme of different targets of anti-cancer drugs within a cell. As you could see, this is a typical cell with a nucleus, rough endoplasmic reticulum, Golgi apparatus, mitochondria, smooth endoplasmic reticulum, certain vesicles here and there, and then the cytoskeleton elements here and here, yeah, and cell surface receptors present here. Now this cell, as part of its fellow cells in the tissue, would be served by the blood vessel. Asparaginase is a drug which inhibits, which actually inactivates the asparagine and does not allow it to be used by the cancer cells. Asparagine is an essential nutrient, and its lack would lead to death of the cell. Immunoglobulins and tyrosine kinase inhibitors, as well as hormones, they have their receptors, respective receptors, usually at the cell surface or sometimes also within the cell cytoplasm. Zinc alkaloids and hexanes, they target the microtubules, and they are known as the microtubule poisons. Antimetabolites are a group of drugs which inhibit the folate metabolism or inhibit the incorporation of correct nucleotides into the growing chains of DNA and RNA. Whereas anthracyclines, bleomycins, and etoposide, they are different compounds which target DNA one way or the other. For example, anthracyclines are intercalating agents. Bleomycin produces scissions of the or nicks in the DNA strands, whereas etoposide is a drug which inhibits the topoisomerases. Alkylating agents and platinum compounds are a group which cross-link the DNA strands or RNA and then inhibit the DNA replication and thereby cell growth and differentiation. To choose drugs to treat cancer, we need to understand their efficacy during different phases of cell division or cell cycle. A cell cycle starts with G1 phase, which is growth 1, then followed by S phase, followed by the second G phase or G2 phase, and then ultimately the mitotic phase or M phase. And then the cycle continues. Some of the cells, they would remain quiescent, do not enter the next cell division cycle, and they only enter the cell division cycle upon the need or the stimulus, appropriate stimulus to drive them into the active cell cycle. These arrowheads show the checkpoints, biological checkpoints, which are important in the progression of the cell from one phase of the cycle to the other. We have certain drugs which are cell cycle non-specific. That means they are effective in any of these 5 phases of the cycle. For example, alkylating agents. We have another bunch of drugs which are cell cycle specific. That means once the cell is in active cell division from G1, S, G2, and M, only then these drugs would be effective. And in G0, they remain largely ineffective. For example, anthracyclines. And then we have a third group of drugs which are called as the phase-specific drugs. These are effective only during a specific phase of the active cell division. For example, RSC or cytosine arabinoside, 6-mercaptopurine, methotrexate, and others. They are the drugs which are most effective during the synthetic phase, where the DNA synthesis is actively taking place. On the other hand, vincristine and paclitaxel, they are the drugs which inhibit the mitotic cell division. Another related concept is to understand the fraction of those cancer cells which are in active growth, or simply growth fraction. Generally, actively dividing cells are around 5 to 10% of the total cell mass. These are problematic, the fraction of the cell, because they grow and replicate and produce the bulk in the mass. But they are susceptible to chemotherapy because they are actively dividing. On the other hand, the quiescent cells, which are in G0 phase, they serve as the reservoir cells and potentially are problematic. Why? Because they are not much susceptible to chemotherapy, hence difficult to eradicate. To eradicate these cells, one has to induce these cells, the quiescent cells, into the actively dividing cell pool, and then the chemotherapy would be effective. The third fraction of the cell is of the cancerous population could be non-dividing cells, which have lost the ability to divide. These are least important for the sake of treatment prioritization. Since different cancers have different growth fractions, it is important to understand what could be the best strategy to kill the cancer cells. However, the cancer cells are not killed all at once. Some give way earlier than others. This leads to the hypothesis of log kill. In this slide, you could see that along x-axis, this is the time, there's a timeline in months to years, and along the y-axis is the number of malignant cells. 10 is power 2 and 4 and 6 and 8 until 12. Most of the times, this is a general description, most of the times the diagnosis is evident somewhere here when the cell population grows this much, whereas the symptoms do appear around this phase, and once the symptoms appear, if not treated, the cancer is rapidly lethal. However, if you remove the cancer mass in a solid organ, for example, in a solid organ tumor, and follow it up with chemotherapy or radiotherapy or both, this is called as the vigorous, that means a vigorous treatment of the cancer. This will ultimately reduce the cell populations with each subsequent cycle of the chemotherapy or radiotherapy, and over the period of few months, the cancer cell population would decline and becomes undetectable. But then you have to follow it up, maybe for 3 years or 5 years or so, to ascertain that the cancer does not recur. On the other hand, if surgery is not possible, for example, only the chemotherapy and other associated modalities are the treatment options available. This will lead ultimately to the decline in the cell population, but will take longer time and much more number of cycles as well. On the other hand, once the treatment option is only palliative, that means it either could be partially treated or could be palliative intent of treatment, then the goal of the treatment is just to improve the survival and gain some more months or a couple of years before the person ultimately dies. And look at that, although there is a reduction in the cancer cell, it tends to overpower and then ultimately would lead to death of the individual. Now let's review modes of action of different chemotherapeutic drug groups. This slide shows how folate metabolism could be targeted. Para-aminobenzoic acid and pteridine together form dihydrotauroic acid, which ultimately becomes folic acid. Folic acid is reduced to dihydrofolate and then tetrahydrofolate. These are the enzymes. Tetrahydrofolate is used to synthesize purines, certain pyrimidines, and certain important amino acid regeneration, for example, methionine. Ultimately, all these functions lead to healthy cell replication and growth. Methotrexate is an anticancer drug which inhibits dihydrofolate reductase here and then leads to inhibition of DNA replication and cell growth and division ultimately. The most logical adverse effect of disrupting folate metabolism is bone marrow suppression or myelosuppression, which responds only to replenishing the folate in this case. However, it will also bring back the cancer cells. Here we have an alternative. Which is called as the folinic acid or leucovorin, famous brand name. Normal cells are relatively quicker to respond to leucovorin than the malignant cells, and hence they replenish quicker. Now let's see in an animation. Dihydrofolate and tetrahydrofolate, you know them already. Tetrahydrofolate then is converted into different intermediates like methylene, methinyl, and formyl tetrahydrofolate. Some of them are bidirectional and some of them are only unidirectional flow of the reaction. Please remember that all these compounds or intermediates are actually forming a part of different metabolic reaction cycles or pathways. As we know, methotrexate would block the conversion of dihydro to tetrahydrofolate, so the body would lose all these intermediates of tetrahydrofolate. However, if you administer folinic acid, you will regenerate methinyl tetrahydrofolate directly, and this could be changed into formyl tetrahydro and methylene tetrahydrofolate alternative. Thus, folinic acid rescues the myelosuppression in the normal. Another important mechanism of action of metabolite anti-cancer drugs is to function as a false or pseudo nucleotide. If an imposter nucleotide is administered, this is called as a pseudonucleotide. Normally, nucleotides are synthesized as either purines, which include adenosine and guanosine, or pyrimidine, which include cytosine, thymidine, and uracil. The nucleotides are then polymerized to form a long chain of DNA or RNA. If a false nucleotide is administered, it can get through the normal system and inhibit DNA or RNA synthesis at a certain point. For example, the uracil is converted into thymidine, which is required for DNA synthesis. And this reaction of conversion of uracil to thymidine is under control of thymidylate synthase enzyme. 5-fluorouracil, which looks like uracil, is an imposter nucleotide or pseudonucleotide, which when administered would ultimately inhibit this enzyme. Once it is part of the cycle, it will inhibit this enzyme and ultimately deprive the cell of thymidine, leading to thymidine-less death. Let's see, now once we have seen different antimetabolites, such as methotrexate and fluorouracil, let's see an interesting example of a collaborative or synergistic effect of two antimetabolites. Here you could see folate is ultimately converted to tetrahydrofolate, and here is the precursors of the thymidine, DUMP and DTMP. And this DTMP is the active nucleotide which takes part in DNA synthesis. This conversion is actually under control of tetrahydrofolate, which takes part to regenerate the enzyme responsible for the synthesis of DTMP. The enzyme is thymidylate synthase or synthetase. So fluorouracil would inhibit this thymidylate synthetase, whereas methotrexate would inhibit dihydrofolate reductase. Together, they have got a profound effect and help in controlling the cancer cell growth. Another important method of killing cancer cells could be irreversibly cross-linking DNA or RNA strands, although it has got its own long-term effects. Here you could see an alkylating agent, the metabolite of cyclophosphamide. This is phosphoramide mustard, and also compare it with cisplatin. Now look at that. Both these have got halogenated ends here, which function as if they are two hands, and they can grab hold of two strands of DNA or a DNA strand or an RNA or something like that. This is a DNA strand, and this is the complementary DNA strand. Phosphoramide mustard is particularly important in cross-linking the DNA where there are two adjacent guanosine nucleotides, G to G. This could be between the opposite strands or within the same strand. In this case, this is called as the interstrand, and here it is called as the intrastrand cross-linking. On the other hand, anthracyclines intercalate between two DNA strands, and thus both these drugs, that is the alkylating agents as anthracyclines, they inhibit the DNA replication. But anthracyclines does it non-covalently. What is intercalation? Intercalation means physical setup of the guest particles between the two host strands of the DNA. Intercalation is usually reversible thermally or chemically because this is not a covalent bonding. It's just a physical bonding and has got weak bonds. So these guest particles could be overthrown by some mechanism which doesn't operate in the living tissue. So I'm sure that you know that DNA replication would lead to supercoils and consequent inhibition of further replication. If a group of enzymes called topoisomerases weren't there, this is a DNA strand, and this is the complementary DNA strand. Topoisomerase type 1, if there is a supercoil, breaks one of these strands and rotates it around the axis of the other intact strand so that the supercoil is relieved. On the other hand, topoisomerase 2 does the job by simultaneously cutting and religating both strands. The topocyte functions by inhibiting this mechanism. Ultimately, it's cell division which produces new cells. So, as we know, microtubules play an important role in cell division. Could they serve as a feasible anti-cancer target? Let's see. Active cell division is characterized by following phases: prophase, metaphase, anaphase, and telophase, followed by cytokinesis. Please note that the microtubules play an important role during metaphase and anaphase because microtubules form the mitotic spindle which helps to pull the chromosomes to the opposite poles. This animation shows how something attached to microtubules moves along its course. And just focus on the flashing, white flashing point and to see how really it moves with addition and subtraction of the alpha and beta tubulin units. That's yeah. Now look at the white flashing white point as it moves along the entire length of the microtubule because more is added here and subunits get out of the tubular length from this side. That's why this end is also called as the positive end and this one is also called as the negative end. No wonder we can inhibit microtubule dynamics and thus inhibit cell division. The same microtubule responsible for chromosome pulling during the mitotic cell division. New microtubule subunits are added from the positive end and subtracted from the negative end. Please note that at both ends there would be some addition and some subtraction, but it's the net addition at one end and net subtraction at the other, and that's why we call them positive and negative ends. Zinc alkaloids, vincristine, vinblastine, these are the vinca or paravincal alkaloids. Vinca alkaloids inhibit the polymerization at the positive end, whereas taxanes stabilize the negative end so that the subtraction of the microtubule subunits doesn't take place. This leads to redundancy, and this leads to inability of anything, for example, attached here to move in this direction, because that portion now is stabilized, no more subtracted. So this portion won't move anyway any further in this direction. Recent research has explored the role of immunity against cancer and has led to development of a number of molecules which either modulate important steps in immune reaction to cancer or block a cell growth signal. So let's explore immunoglobulins as the anti-cancer agents. As you can see, this is a schematic diagram of a cell membrane, and this is an immunoglobulin, this is an tyrosine kinase coupled receptor, which has got an extracellular domain, a transmembrane domain, and an intracellular domain coupled with tyrosine kinase. The growth factor of this receptor, when it arrives, binds to its respective receptor. This activates the enzyme tyrosine kinase ultimately, and there is a further downstream growth signaling which is set in, and ultimately there is growth and differentiation of the cell. If this growth factor is either inactivated, for example, with bevacizumab for vascular endothelial growth factor, or if an immunoglobulin prevents its binding to its target receptor, for example, trastuzumab for HER2 and cetuximab for epithelium-derived growth, EGRF. Another way, if these dynamics of the receptor and the growth factor is disrupted, the ultimate kinase activation and downstream growth signaling is affected, is no more available, inhibiting the cell growth and differentiation. Another related drug is imatinib, which is not an immunoglobulin. It gets into the cell and directly binds to the kinase rather than disrupting the inhibition at the extracellular level. It's interesting to note that how research evolved immunoglobulins into types which resemble now very much with the human immunoglobulins, and hence are better tolerated than the earlier foreign immunoglobulins. Look at the types of immunoglobulins. This is an all-mouse origin, whereas this one has got a more human structure, but still the variable region is as such is of mouse origin, so it's a chimeric antibody. Here you could see in the variable region, only the clone regions of the CDRs are the ones which are, the ones which are from the mouse. Otherwise, is almost completely human in origin. And here is the complete all-human immunoglobulin. Now let's review some important general aspects. As you could see, most drugs are anti-proliferative in terms of the cancer chemotherapy. Hence, they inhibit the growth, but they don't have a really good or very effective effect on the differentiation or spread. Tumors with the large growth fraction, you can use specific agents, cell cycle specific agents like mitotic spindle poisons. Whereas with the tumors with the smaller growth fractions, it's better to use non cell cycle non-specific agents like cyclophosphamide. The dosing in cancer chemotherapy is intermittent rather than continuous in order to avoid resistance, because cancer cells are very smart in that quality, and they very often develop resistance against the drug. Another reason why another way of avoiding the resistance is to combine different drugs, the drugs which are different in terms of their mode of actions and their spectrum of the side effects. Anyhow, success depends on the type and stage of the cancer at the time of the diagnosis, and development of the drug resistance, and overall health of the patient, whether he or she could take the chemotherapy, the burden and the stress of the chemotherapy or not. Because remember, these drugs are poisons not only for the cancer cells but also for the normal cells, and it's just the differential killing which makes the difference. Let's also review important disadvantages of current chemotherapy. As you could already see, indiscriminate destruction is the approach rather than the reversal of pathology. There is a high likelihood of adverse effects because of the low selectivity. But this is mostly dose dependent, of course. A number of anti-cancer drugs are also used as an immunosuppressive at low doses. Just to remind you that this adverse toxicity, the adverse effects are mostly dose dependent. For example, cyclophosphamide could also be used in certain autoimmune disorders like rheumatoid arthritis. But at the same time, used in heavy doses could be anti-cancer drug. And as we already figured out, total elimination of the mutant cells is not possible. Hence, you repeat the doses all the time. And then there is a chance, very bright chances of development of resistance in the mutant cells. Let's also review some adverse effects and the strategies to treat them. If you could see on this far left column, here we have listed certain tissues like bone marrow, urinary tract, liver, and so on. And here are their respective adverse effects. Many drugs, they can produce myelosuppression. A lot of them are nephrotoxic, and cyclophosphamide could also produce hemorrhagic cystitis. Many drugs of them are hepatotoxic. Some of them are ototoxic as well. And since any rapidly growing tissue or cell type is affected, one encounters hair loss or alopecia, as well as skin and mucosal damage if he or she is put on the chemotherapeutic agents. One important adverse effect is the nausea, vomiting, and sometimes diarrhea as well. And this is related directly because of the direct irritation of the vomiting center, chemoreceptor trigger zone in the brain, or causing a mucosal damage. Sterility because of the gonadal germinal layer epithelium is also very much common. And then equally important is the ability to inflict damage to the growing fetus or teratogenicity. If given in children, many of these drugs, since they inhibit growth, so they result in stunted growth. And because many of these drugs would also lead to widespread killing of the cells, they are a source of production of uric acid and can lead to a gouty attack. Now here are the treatment options. For the myelosuppression, autograft is a recommended treatment. Although gene therapy has also been, devised and formulated, but not so much successful. For the nephrotoxicity, you could keep the patient hydrated. For prevention of hemorrhagic cystitis, Mesna is an agent, is a drug which is given together with the high-dose cyclophosphamide therapy, and Mesna then inactivates the phosphoramide mustard and the acrolein, which is another metabolite of cyclophosphamide, and hence Mesna can prevent the toxicity exhibited by high-dose cyclophosphamide therapy to the urinary tract epithelium. As you could see, the treatment of hepatotoxicity, autotoxicity, and skin problems is supportive only. For nausea vomiting, you need to use strong antiemetics, primarily the ones which block the serotonin receptors. For the sterility, again, the supportive measures, and for the prevention of teratogenicity, strict contraception is the rule. Nothing is so far available for the standard growth very much. And for the gout, you could use allopurinol to block the formation of the uric acid. I hope you enjoyed being with me today, and thank you for your interest in watching this lecture. Thank you very much.